Okay, last Dr. Ben update of the day. Let's go through the last couple of papers super fast and superficially. Yeah, because they're all pretty easy to get. So, again, these are preprints, not fully part of the official scientific record yet, but they're likely to be in some form pretty soon. All right, so the first one is looking at different antibody tests and comparing them. And it turns out that all of the 5 tests that they are comparing, including a couple of their own in house tests, are about the same in terms of how they score. So the good thing is that all of these tests get 93% or more positive. So rather, they detect 93% of people that are actually sick once you get to 10 days after they were detected positive by the PCR test. And we know the PCR test probably doesn't work until something like 7 days after they were infected in the first place. So we're talking something like 17 days yeah, from the actual point where they're infected, and probably something like 12 days or 13 days since the point where they could infect other people. So totally useless for controlling the infection, but very good for actually formally proving who was infected, even after their body has dealt with the virus and got rid of it. And so, yeah, there we go. At about 5 to 9 days, so think 1 week plus or minus 2 days is I think what they're going for there, they could detect about 2/3 of the cases, and that was sort of just barely. So the antibody tests do not work on a very fast time scale. But yeah, in a long-term sort of situation where somebody's had the virus for a long time, or where somebody has had the virus and recovered, yeah, they should be able to tell. So boom, done. All right, next one. Next one is another one of these. Oh my gosh, there's so many single cell profiling studies, and it looks at cells in the eye. And it turns out there are some cells in the eye. So there are cells in the conjunctiva, which is this membrane in here that gets all red when you have conjunctivitis. And there are a couple of corneal cells that actually have the 2 proteins we need, ACE2 and TMPRSS2. And some of these also have the other protein that can help out and do the job of TMPRSS2, but maybe isn't as good as it. It's called cathepsin type L. And so, yeah, it looks like, at least in theory, you should be able to be infected through the eyes. But I guess the nice part, if there is one, is that most of the cells they looked at in the eye, they looked at, I don't know, 30, 40 different cell types. Most of them were missing one or even both of those proteins. So it's not going to grow in the eye rapidly and forever, but theoretically a person could get infected there. So now we know. All right, boom, another one done. Next one is, oh yeah, next one is showing that the spike protein, so the one on the outside of the virus, is able to bind to heparin sulfate. And so heparin sulfate is this sort of sugary molecule that's going to be on the outside of cells. And so this is showing that the spike is generally able to get close to cells. It's able to bind something that's bound to cells. And this is probably a preliminary step. It's sort of homing in on its target, which is ACE2 that's next to TMPRSS2, or maybe cathepsin L. Those are our things that we have to have for entry. But it's showing they can stick to heparin sulfate as well. There's a lot of coronaviruses that can do this. They can bind to some extent. And this is, we think, maybe why coronavirus is so big. It's got a little part to bind ACE2, which is the part that pops up here. It's got other parts that are going to bind something like heparin sulfate or may interact with other proteins or sugars. It's got other parts folded up inside that are kind of like a transformer that are going to unfold and hook a cell and fuse the virus with it. Yeah, really neat stuff. Yeah, it makes for really great animated videos. There's a couple of those on YouTube that show how it works. So there we go, heparin sulfate, great. And last one is, oh yeah, I remember now, is showing that the, so inside the nose, there's a little patch where you have all your like nasal, all your scent receptors, and these are called neuroepithelial cells. Bleh, epithelial cells, there we go. Yeah, and so they are going to be cells on the surface of the inside of your nose, but they're also going to be connected up to neurons. And they are the ones that are going to pick up scents and actually signal through those neurons and say, oh, that smells like, warm cookies or whatever. Yeah. And so there's a large proportion of these seem to have high amounts of ACE2 protein, and so they are potentially infectable. Yeah. And so this could potentially explain some of these reports, which are still not super confirmed as far as I know, that people lose their sense of smell and taste as part of one of the symptoms of COVID. So that could explain it. If the virus is actually growing in those cells that are supposed to be smelling, then they're not going to work anymore, and then you're not going to be able to smell. So this is not proof that that actually happens. This just says that, oh, the cells have the right things so that the virus could grow there if it got there. And so, yeah, it may happen. It seems reasonable anyway. Thanks very much. Dr. Ben updates. Yeah, 4 papers in one update. That's pretty good. Talk to you later.